In today's lecture, we learn about the basic breeding approach to any crop improvement, which is selection. Isolation of desirable plant types from the population is known as selection. Selection is one of the two fundamental steps of any breeding program. And the two fundamental steps of any breeding program are creation of variation and then selection. However, the effectiveness of selection primarily depends upon the degree to which phenotype reflects the genotype, that is, the extent to which the genetic background of a particular species is represented by the phenotypic characters. There are basically two agencies of selection involved in carrying out selection. One is a natural selection and the other one is the artificial selection. Natural selection is nothing but the selection by nature itself. Artificial selection is nothing but the selection which is carried out by man. Though both may complement each other, in some cases they are mostly opposite in direction since their aims are different under the two conditions, that is nature and then domestication. The crop species under domestication were exposed both to natural and artificial selection. Before domestication, crop species were subject to natural selection. The basis for natural selection was adaptation to the prevailing environment. And the crop species under domestication, that is after domestication, man has knowingly or unknowingly practiced some selection in order to isolate superior genotypes. But in modern plant breeding methods, natural selection is of little importance, whereas artificial selection plays an important role. There are three basic principles of selection given by Walker in the year 1969 to have a better knowledge about selection. And these principles goes like this. The first one is selection operates on existing variability. That is, selection acts on the existing variation present in the population and it cannot create any new variation. And the second principle is that selection acts only through heritable differences. Hence, the variation among individuals to be selected must be genetic in nature since it is the genetic variation that tends to close the gap between phenotype and genotype. Environmental variability cannot be of any use under selection. That is, it is the genetic variation which actually passes on from one generation to the another generation. And the third principle says that selection works because some individuals are favored in reproduction at the expense of others. That is, some individuals tend to become superior to others for some or other traits desirable under domestication. These superior individuals are retained for reproduction, while the rest are discarded under selection. Selection has two basic characters. They are selection is effective for heritable differences only, and selection does not create any new variation and it utilizes the variation already present in a population. And there are two basic requirements for selection to operate. They are variation must be present in the population and the variation present in the population should be heritable. And here we are going to see about a special term called selection intensity, which is the percentage of plants selected to be advanced to next generation from a population. That is, the number of individual plants selected from the whole population which are to be advanced to the further generation. And we basically have two types of selection, which are namely mass selection and Pureline selection. Mass selection is the earliest method of selection And it has always man has always practiced mass selection consciously or unconsciously from the time of domestication itself. Mass selection refers to a method of crop improvement in which individual plants are selected on the basis of phenotype from a mixed population, and the seeds are bulked and used to grow the next generation. And now, if we see the procedure for evolving a variety by mass selection, what happens is that during the first year, large number of phenotypically similar plants having desirable characters are selected, and the number of plants selected may vary from few hundred to few thousand. The seeds from the selected plants are composited, that is bulked, and are raised in the next generation. In the next generation or the second year, the composited seed is planted in preliminary field trials, that is, initial field trials for assessing the characters, along with the standard checks. And here, the variety from which the initial planting material is selected was also used as check. Phenotypic characters of the variety are critically examined and evaluated. And from the third year to the fifth year, variety is evaluated in coordinated yield trials at several locations, which means the variety is evaluated simultaneously at several places. For 1 year, it is evaluated in an initial evaluation trial, and those varieties which are found superior in the initial year are promoted to main field yield trials for the next 2 to 3 years. And in the seventh year, those varieties which have been proved superior in the main yield trials with respect to the production or any quantitative trait or qualitative trait is multiplied and released after giving a suitable name. And this is the procedure for evolving a variety by mass selection. And next, if we look into the merits and demerits of mass selection, they can be explained like this. First, if we go with the merits, mass selection can be practiced both in self and cross pollinated crops, and there is no control over pollination. The varieties developed through mass selection are more widely adopted than pure lines. Mass selection retains considerable variability, and hence further improvement is possible in future by selection. Mass selection also helps in preservation of land races. Mass selection is useful for purification of pureline varieties. And mass selection is also useful in the improvement of characters governed by few genes with high heritability. And mass selection in turn is less time consuming and also less expensive. If we see the demerits of mass selection, varieties developed through mass selection are not uniform. And since there is no progeny test done in mass selection, the genotype of the selected plant is not known. Progeny test is nothing but evaluation of the performance of a particular individual on the basis of the performance of its progeny. And the next demerit is, since mass selection is based on phenotype and no control over pollination, the improvement brought about is not permanent. Hence, the process of mass selection has to be repeated now and then. Characters which are governed by large number of genes with low heritability cannot be improved by mass selection. And mass selection cannot create any new genotype, but it utilizes the existing genetic variability. And if you see the achievements of mass selection must have been used by prehistoric man to develop the present day cultivated forms from the wild parents. It was also used extensively before the pureline selection came into existence. And few of the achievements are Dharwad American cotton variety and TMV1, TMV2 groundnut varieties, Pusa Moti, Bajapuri, Jamnagar Jaint, AF3 varieties of Bajra, RS1 of sorghum, SLO13, MTO15 of rice, K122 of potato are all the varieties which are developed by mass selection. And now, if we see the next method of selection, that is pure line selection, which is mainly practiced in case of self-pollinated or autogamous crops. A pure line is nothing but a progeny of a single homozygous plant of a self-pollinated species. And all the plants of a pure line have same genotype. In pure line selection, a large number of plants are selected from a self-pollinated crop and harvested individually. Individual plant progenies from them are evaluated and the best progeny is released as a pure line variety. And if we see the genotype of pure lines, all the individuals within a pure line have identical genotype. As the parent from which the pure line was derived is homozygous and is self-fertilized. Any variation present within a pure line is solely due to the environment. Variation present within a pure line is due to environment and it is not heritable because it is caused due to the external agency and not the internal genetic fluctuations. And pure lines become genetically variable at times with time. What are the reasons? It is mainly due to either mechanical mixtures or natural hybridization or mutations. That is pure line selection. Pure lines developed by pure line selection may become genetically variable with time because of mechanical mixtures or natural hybridization or mutations. And now, if we see the procedure of pure line selection, initially during the first year, about 200 to 3000 plants are selected for superior phenotype and a superior phenotypic character. And then these are raised in the first year and individual plants are subjected to harvesting wherein seeds are harvested from each plant separately. And in the second year, the seeds harvested separately are planted and undesirable progenies are rejected. And in the third year, those progenies which are selected in the second year are planted in preliminary yield trial, that is primary yield trial, and then inferior ones among these are rejected. From the fourth year to sixth year, coordinated multi-location yield trials are conducted along with suitable checks. That is multi-location yield trials. Yield trials at different locations are conducted simultaneously, and disease resistance and other quality tests are done along with the yield trial. And in the seventh year, those varieties or those individuals which were found to be superior with respect to the characters under test were selected and are released as a new variety, and seed is multiplied and is bigger and it is sent for distribution. And this is a procedure for developing a pure line. And the main difference between pure line and mass selection in this procedure is that in pure line selection, after every generation, the seed is collected separately from each plant and it is not composited or bulked, and that seed is individually raised in the next generation. And in pure line selection, strict self pollination is also done, which is not the case with the mass selection where there is no control over pollination. And now, if we see into the applications of pure line selection, the pure lines developed by selection can be directly released as varieties, or they can be used in hybridization program as parents, and they can also be used in the improvement of local varieties. That is, pure line selection can be used in the improvement of local varieties in order to induce homozygosity in the individuals. And then pure line selection can be operated on introduced varieties, and mutations can be conducted on the developed pure lines, or those mutated individuals developed through mutation can be subjected to pure line selection. And the next application is that selection for new characters in pure lines can be done. Those pure lines which are released as varieties can once again be subjected to pure line selection for the selection of new characters in them. And if you see the advantages and disadvantages of pure line selection, first, if we see into the advantages, pure line selection achieves maximum possible improvement over the original variety, and pure line varieties are extremely uniform. They are therefore preferred by farmers and also consumers, in contrast to the less uniform variety developed through mass selection. Due to this extreme uniformity, the pure line is easily identified in seed certification programs. Now, if we go with the disadvantages of pure line selection, the varieties developed through pure line selection generally do not have wide adaptation and stability in the production that which is possessed by local varieties from which they are developed. And pure line selection requires more time, space, and also the yield trials which are adopted are expensive in comparison to the mass selection, and therefore breeder has to devote more time to pure line selection than to mass selection. And the improvement in pure line selection is dependent on the genetic variation present in the original population, and no new genetic variation is created in pure line selection. And so far we have seen about what is selection, its principles, characters, requirements, selection intensity, types of selection, mass selection, pure line selection, along with their procedures, applications, advantages, and disadvantages. Thank you.